Aloha, and welcome to Unit 2, Organization of Matter. So just to introduce you to this chapter, the overview of this lecture is going to be about the introduction of the language or the nomenclature and the basic concepts of chemistry. We're also going to talk about the organization of matter, so how matter is organized into atoms and how those atoms bond to each other to be able to make molecules and what those molecules do with other molecules, discuss how chemical bonds form, different types of chemical bonds, ionic versus covalent, and also talk about chemical reactions and how those chemical reactions can occur. And then, last but not least, we'll be comparing and contrasting the difference between organic and inorganic compounds. Now, some of the basic principles of chemistry. Number one, chemistry is the science of the structure and the interactions of matter. Matter is defined up as anything that has mass and takes up space. And mass is the exact amount of matter that a substance contains, whereas weight is the force of gravity acting on mass. So weight is going to be m times g. All right, so we have three major forms of matter. We have solid, liquid, gas. There's also plasma, but we're going to ignore that for the purposes of this class. And all forms of matter are composed of chemical elements. Now, an element is a specific type of, gets all the way down to atom, and atom is the smallest unit of, and that element that retains the properties of that element. And elements have specific chemical symbols, and they have different amounts of protons and neutrons and electrons, but I digress. So some of the elements that you may see in this class include oxygen, which is depicted as O, carbon, depicted as C, hydrogen, depicted as H, and nitrogen, depicted as N. And those guys together are going to be the major elements. That's about 96% of the elements in the body. And the trace elements, all of the lesser elements combined, are about 4% of the total. But I'm sorry, all of the lesser elements are about 3.6, so about 4% total, and the trace elements are about 0.4% of our total. So we have a specialized set of elements that would come in very, very low concentrations. But for the purposes of this class, we're probably only going to be talking about oxygen, carbon, hydrogen, and nitrogen. And these elements are going to be responsible for making up most of the biomolecules in the majority of our bodies. Now, atoms are going to be the smallest unit of matter that retains the properties and characteristics of whatever the element that we're looking at is. And atoms have subatomic particles, so you might think that the atom is the smallest that there is, but you'd be wrong. It does get smaller than an atom, but once you get down to the electrons and the protons and the neutrons, that's what all atoms are made up of. So there's no way to tell amongst atoms except by the number of protons, electrons, and neutrons inside them. So inside the atom, we have two major regions. We have the nucleus. The nucleus is the center. And then we have the electrons, which circulate around the outside. And this here is shown as the electron cloud model. It's more like a bubble or a film of electrons that race around the outside. But we also use the electron shell model when we're going to talk about how electrons interact with others. So this depicts them kind of in motion, just running around all over the place. And this depicts them statically, in which case we're able to count and use them and measure. So for everything that we're going to be doing for the purposes of this class, we'll be using the electron shell model, which is a little bit less realistic, but is a little bit easier and simplified for us to be able to work with. One of the other things to note here is that protons are going to be positively charged. That's why there's this little positive here. Electrons here are negatively charged. Neutrons are neutral, no charge. So an atom walks into a bar and says, "I've lost an electron." And the bartender says, "Are you sure?" And the atom says, "I am. I'm positive." Waka waka. But that's because the electron is negative, so it's lost the negative charge. Anyway, the atomic nucleus is going to be where the region where the protons, again these are positively charged, and neutrons, again neutral charge, are going to reside. And both protons and neutrons have weight, whereas electrons, for the purposes of this class, do not. So each proton and neutron is going to be 1, and each electron is going to count as 0 in terms of the atomic weight. And the atomic weight is going to be responsible for the atomic number of the so the atomic mass here is 12, and the atomic number is basically half of that, and that's the equivalent of the number of both protons and neutrons. Again, these are generally going to be equal, same amount of protons as neutrons in the number of in the nucleus of the atom. All right, so neutrons, again, as I mentioned, they're approximately 1, protons approximately 1, electrons approximately 0. For the purposes of this class, we're not going to round out all the way out to those decimals. Just some more definitions. An ion is any atom that has lost or gained an electron. So it's going to be positively charged or negatively charged, depending on whether it's lost or gained one. And a molecule is any time that we have two atoms coming together that are sharing an electron. So they're not just bouncing off of one another, they're actually interacting. And when we have molecules that are comprised of multiple elements, that's called a compound. So it's a substance that can be broken down into two or more different elements. So anything that's not going to be just a bar of gold, for example, but will be an alloy or have more than one element involved in it, that's considered to be a compound. Now we have different types of chemical bonds. Basically what happens is chemical bonds occur when atoms come together and are attracted to one another. And we have these different shells that are surrounding the atom. This is the first shell. This is the outer shell in this case. There could be multiple levels, but for the case of carbon, this is the outer shell, and it's called the valence shell. Now, you can picture these like cartons of eggs. They want to be full cartons of eggs. In this case, the carton of egg number would not be 12, it would be 8. So this outer valence shell here has 4 and wants to have 8. I want to take a digression here for a little bit just to demonstrate here. The atomic number is 6, but those first 2 electrons are going to take up that first shell. So really what we're looking at is 4 electrons that are able to interact. So even though there are 6 electrons, these 2 are happy and they're not going to react. And these guys are going to want to find someone to pair up with. This is incidentally what makes carbon unique, because carbon, given that it has 4 electrons, is able to make 4 different bonds with 4 different other atoms. So it has a diversity factor, basically, in all these biomolecules. It's going to be able to make a chain, a nice carbon backbone. And again, the way that these atoms interact with other atoms is defined by the amount of electrons that are found out here in what we call the valence shell, or the outer shell of the atom. Now, anytime an atom has picked up or released an electron, it goes from being an atom into an ion. An ion is a charged atom that has either picked up or released an electron. And as I mentioned previously, these outer shells want to be, I called it a full egg carton, but it wants to have 8 in this one. So if you want to have 8 and you only have 1, you have 2 options. You can either get rid of this one, go down a shell, and then you have a happy 8, or you can pick one up. Well, you can pick up 7, I guess, right? Or you could share one. In this case, it's going to want to release one because it's going to be a lot easier to release one than it is to pick up 7 than it is to share 7, right? So it's going to donate that electron off of this atom to become an ion. When you lose an electron, remember electrons are negatively charged, that means the ion is going to become positively charged. In this case, this is chlorine. Chlorine has 7 valence electrons, and it's going to want to pick up 1 because it wants to have 8. So it's a lot easier to pick up 1 than it is to release 7. And so it's going to accept that electron from a nearby atom. In this case, it's sodium, so sodium wants to donate 1, and chlorine wants to accept 1, so that's a very nice, happy interaction. And it leaves sodium a little bit positively charged, right? It gives it a positive charge, and it leaves chlorine positively charged. These are now ions. Now, these ions kind of want to stay close to one another because now they have a magnetic attraction. They have a positive charge and a negative charge that keeps them not only next to one another but oriented in a certain direction, and that's why we're going to have a nice crystalline structure of something like table salts. Okay, so let's talk about how atoms interact with other atoms. Now, what we just talked about was an ionic bond, whereby the electron is actually donated from one atom to another atom, right? And then these guys hang out next to each other, and they're both happy because they both have 8. Now I'm going to talk about covalent bonding, and covalent bonding is when electrons are shared instead of just donated from one to another. So for example, hydrogen here, hydrogen has 1, and its valence shell is 2. So everything else that we're working with is going to be 8, but in this case we're working with 2, and since they each have 1, they're going to share, and if they share, now they both have 2. Right? That's a single bond. It's one bond where we're sharing 1 electron. In this case, it's 2 hydrogens, so the structural formula is hydrogen-hydrogen with 1 single bond depicted as one line. The molecular formula is H2. If we look at oxygen atoms, they're also able to bond to one another to make an oxygen molecule. Remember, you only need 2 atoms together to become a molecule. Here, these guys have 6 in their outer shell, and they want to have 8, so they're going to each share 2. That means that they make a double bond amongst themselves, and therefore they both are happy because they have 8 because they're sharing 2 amongst themselves. That's depicted as 2 oxygen atoms. In this case, it's an oxygen molecule with both of them together with a double bond. Here's the molecular formula of that molecule, O2. This is nitrogen. Nitrogen is able to make 3 bonds, so it's actually kind of unique because it can make a triple bond or it can make 3 single bonds. So you can make an NH3 molecule, for example. In this case, we have 2 nitrogen atoms coming together making a nitrogen molecule, and they're sharing these 3 that forms a nice triple bond, 2N, and then with the triple bond in between indicates that we have a structural formula where these guys are sharing a triple bond, and the molecular formula is N2. Now note that the molecular formula doesn't tell you what kind of bonds you have. You have to kind of draw it out in your head, although chemists can see this pretty much off the top of their head. Okay, so here's what's unique about carbon, right? Carbon has 4. That means that it can share 4 different electrons with 4 other atoms. So carbon can bond 4 hydrogen atoms, for example, and make CH4 or methane. Right? Each of the hydrogens is happy because it's sharing 1 electron with carbon. Carbon's happy because it's sharing its 4 with hydrogen's 4, so it has 8. Oxygen can share 2, but because we have 2 and we actually are sharing it out of 8, we have an abnormal shape. Additionally, another reason we have an abnormal shape is because hydrogen has 1 shell and oxygen has 2 shells. And so what that indicates is that this shell only needs 2, this shell needs 8. This shell is going to be a lot smaller than that one. So if you would picture that you were an electron that was being shared between oxygen and hydrogen, you would spend a lot longer orbiting this much larger atom than you would orbiting the smaller atom, and that would give us partial positive and partial negative charges. So that means that water is a really unique molecule because it has a partial negative charge on the oxygen because it hoards the electron a little bit longer, and a partial positive charge on the hydrogen because it doesn't get to orbit hydrogen for as long, so the electron's not going to be there as long. That's going to give us an oddly shaped molecule here. Here's the OH, or H2O, and you're going to see it depicted kind of like a Mickey Mouse sometimes, right? It's going to kind of look like Mickey Mouse with Mickey Mouse ears, as you also know it as H2O. So this is going to be the molecular formula, and this is going to be the structural formula. And what's important to note is that we have partial positive and partial negative charges. That gives this molecule what we call polarity. So water is a polar molecule. All right, so let's talk about hydrogen bonding for a second. So hydrogen bonding is when atoms from other molecules associate. So here we have atoms from water molecule A and water molecule B. Here we have partial positive charges on this hydrogen and partial negative charges on this oxygen. That's going to give an orientation of water. That's going to mean that we have a little bit of a bond between this hydrogen and that oxygen. This is going to be very important when it comes to like water viscosity, for example, or the ability of water to make long chains to be able to get water all the way up from the roots to the top of the tree, all the way from the roots to the top of the tree. And water is actually going to be very important when it comes to different biological molecules. We'll talk about that in a little bit, but it's able to divide into OH and H, and that's going to allow it to accommodate different molecular reactions that are happening around it. And we'll also talk about that when we get to the pH scale, which is going to show up just a little bit later on in this lecture. But I digress. Let's talk about chemical reactions. So a chemical reaction happens anytime molecules change from one molecule to another. So we're actually going to have a conversion from molecule A to molecule B, and that happens by creating new bonds or breaking old bonds down. So we can take 2 smaller substances, put them together and make a larger substance. We can take a larger substance, break it down into 2 or 3 smaller substances, or we can take something that's about the same size and just convert it by changing where different atoms are bound, or just moving one atom, replacing one atom with another, for example. Anyway, anytime a chemical reaction occurs, we have what we start off with. That's our substrates or our reactants. That's what was there before the reaction occurs. And then we have products, which are the end goal of the reaction, right? That's our ending substances. And for every reaction, there's an equal and opposite reaction. So every time we put A together with B to make C, we also are able to break down C into A and B. So we have equal reactions that can happen each direction. And the conversion of all of these molecules from A to B into C into D, etc., is the metabolism. So anytime we're breaking things down, we are also building things up. And that is what metabolism is. It's the sum of all of the metabolic pathways, the anabolic, which is the creation pathways, and the catabolic, which is the breaking down pathways. Another thing I want to talk about here is the law of conservation of mass. And what that means is that we're going to end up with the same amount of mass after a reaction as when we started a reaction. So we might end up with totally different molecules, but they're going to weigh the same. So for example, here's the reactants. Here we have 2 H2. So this is 1 H2, this is the second H2. That's 4 hydrogen in total. Now if you look here on the right, we have 4 hydrogen in total, so we haven't lost any. Here we have an O2. It's going to be broken down, and we're going to create 2 H2O molecules. Again, we have 2 oxygen atoms on this side as well. So we have conserved all 4 of our hydrogen atoms and both of our oxygen atoms in the new products. So the mass has actually been conserved. Okay, so let's talk about energy for a little bit. So we were just talking about metabolism, and metabolism is the sum of all of the metabolic or energy pathways in our body, right? That's the chemical energy. But we have also potential energy and kinetic energy. So potential energy is stored energy, and kinetic energy is energy of motion. And chemical energy, as we just talked about, is the energy used to convert one molecule into another. And overall, we're going to have total conservation of energy in the universe. Energy is neither created nor destroyed. It's simply converted from one form to another. Now, we do have loss of usable energy from one conversion to the next, so we do end up headed towards what we call entropy, so like chaos. So the amount of usable energy is lost, but the total amount of energy is always the same on one side of the reaction as the other, because energy is neither created nor destroyed. In fact, all of the energy in our entire ecosystem is going to start off coming in from the sunshine. Okay, so multiple different types of chemical reactions you will hear us talk about throughout the course of these lectures. One is a synthesis reaction, and this is when we are taking two smaller molecules and putting them together to make a larger, what we call macromolecule, synthesis. So taking two and making one, but it's larger. Decomposition is the opposite of that. We're taking one and breaking it into two smaller ones. And usually what happens is the larger molecules have a higher biological complexity than the smaller molecules. So when we are synthesizing something, we are making something that has a higher level of biological complexity and probably a higher level of energy involved in it. And when we're breaking something down, we are generally releasing that energy and making two simpler molecules. We also have exchange and reversible reactions, whereby an exchange reaction is where we're exchanging one molecule for another. Most reactions are reversible, but not all. But reversible reactions would be something where you could go, say you're taking A and you're adding B, you combine those together and you create a larger macromolecule we'll call C. If we take C and break it down into A and B, that would be the reverse reaction. We also have oxidation reduction reactions, and we'll talk about redox reactions. These usually happen at the same time, where we have an oxidation of one molecule and a reduction of the other molecule. And this is kind of an exchange that occurs amongst molecules. And redox reactions are actually very common in the body, so we'll talk about them quite a bit later on as well. So this is the moment where I crawl up on my soapbox and I tell you that every vegan who shouts that they only eat organic is actually lying to you, because water is an inorganic molecule, and obviously they are going to be consuming water even in their most, in their least pesticide fruits and vegetables. So just as a side note, when scientists talk about organic and inorganic compounds, it is completely different than when the rest of the world talks about organic versus inorganic. Inorganic simply means that there is no carbon, right? And so they're very simple molecules generally, like water. They're not carbon backbones. That's all inorganic molecules. Organic compounds always contain hydrogen and oxygen, but generally they specifically have to have carbon. Okay, so they're always going to have carbon, but they are always going to have hydrogen and generally oxygen as well. And they are also going to be held together by covalent bonds, so not by ionic bonds. And this is very important because in a biological system, we're going to have water as the solution in most of our cells and in our bloodstream, etc. And ionic bonds rapidly dissolve in water because of those partial charges, those partial positive and partial negative charges add up and dissolve ionic compounds pretty easily, whereas covalent bonds are going to be stronger structurally in an aqueous environment. So. All right, so this is what I was just talking about, how water has a little bit of a partial positive charge and a partial negative charge. This is what's called a polar covalent bond, and it's going to be easy to remember because water is a polar molecule, and polar bears, if you just remember, polar bears live on ice, and ice is made of water, therefore water is a polar molecule. So it's pretty simple to kind of keep that in your head. And why is it polar? What does polar mean? And polar means that it have an unequal sharing of these electrons. So this electron, again here, is going to spend a longer time around this larger oxygen, less time around the smaller hydrogen. That means it's going to be a partial negative charge on the larger oxygen because it has the negative electron for a little bit longer. Negative electron spends less time around hydrogen, so we have a partial positive charge. Again, our structural formula is going to be depicted here with two single bonds, one between each of the hydrogens and the main body of the oxygen. And the molecular formula you may recognize here as H2O. Now, I was just saying that ionic bonds don't do well, ionic molecules don't do well in aqueous solution, and this is exactly why, because water is a really good solvent, and these partial positive and partial negative charges can add up. So it makes it kind of a promiscuous molecule in that all of these partial negative charges can outcompete the negative charge that chlorine provides and draw sodium away. And at the exact same time, these partial positive charges can outcompete the positive charge that sodium provides and steal chlorine away. And so we can end up with hydrated ions This is essentially what happens when you put a tablespoon of salt into your water, right? Your sodium and your chloride ions are going to dissolve into aqueous solution. And that makes it really important for chemical reactions in the body, right? Water is the ideal medium. So not only do what we just talked about, these partial positive and partial negative charges can disrupt ionic bonds, but in terms of covalent bonds, we can have hydrolysis occurring. And I mentioned this previously. It's neat. So we have H2O, which can break down into H plus and OH negative. So water can actually be split in half. And the process of splitting water in half is called hydrolysis, or lysis, meaning breaking in water. And in a dehydration synthesis reaction, which is the opposite of a hydrolysis reaction, water is actually going to be removed from the two molecules and then end up released. So if we had, now as I mentioned, all reactions generally in the body are going to have an equal and opposite reaction. So let's start here on the left. Here we have glucose by itself and fructose by itself. And you can see here that we have an OH hanging and an OH hanging. When the OH here from glucose comes off and the hydrogen from this OH comes off, okay, that oxygen remains, then we can put this O over here kind of like a matchmaker, and this ends up being sucrose. It also is going to release water at that point. That's called dehydration synthesis. Why? Because we are removing, or dehydrating, we're removing one water molecule off of these two molecules and bonding them together. Now they're left holding hands here where this oxygen is, and water is free in solution. That's dehydration synthesis. Now if I walk you through the equal and opposite reaction, right, where we are taking sucrose and catabolizing it, basically, we're doing a catabolic reaction where we're breaking sucrose down into glucose and fructose. And how do we do that? We're going to take water, we're going to split it in half. We're going to end up with one free hydrogen, which is going to bond to this oxygen and come over here. Then we're going to end up with an OH group that's going to bond here. And now we're going to end up back where we started with glucose and fructose. And that's called hydrolysis. Why? Because this water molecule here is split in half. It's hydrolyzed so that we end up with an OH bonding and an H bonding. Okay, I know that seems like a lot to go over, but basically if you just remember that absolutely every anabolic pathway, so anabolic steroids pump you up, think Arnold Schwarzenegger, right? Anabolic pathways build things up, they build things together. And catabolic pathways break things down. And the summation of anabolic and catabolic pathways in your body is called metabolism, right? So anytime that you have an anabolic reaction where you're taking two smaller molecules and putting them together to make a larger molecule, it does that by dehydration synthesis. We're losing a water molecule to come free in solution. But if we're taking a larger molecule and we're breaking it down into two smaller molecules, we're doing that by the process of hydrolysis. Okay, so all catabolic reactions where we're breaking things down from larger to smaller happen by hydrolysis, whereby a water molecule gets split in half and the free OH goes to one side and the free hydrogen goes to the other side. Again, I know that's a lot to take in. You can refer back to your textbook if you need more information. All right, so some other interesting characteristics of water. Water is very important for being able to maintain temperature because it has a really high heat capacity and a high heat of vaporization. And so that allows you to, for example, go sunbathe in the hot sun without actually sending your temperature skyrocketing, because it takes a lot of energy to heat water by 1 degree. Another thing that's very important about water, particularly when we're talking about organs, we were just talking about the pleural membrane in the last lecture, but water is actually a lubricant. It's a major component of all of our bodily fluids, but it also is going to help reduce friction as our organs and membranes are going to slide over one another. Think as the heart beats, for example, or as your lungs breathe, or they're going to expand and contract and expand and contract. And that water that's going to be between those layers is going to help those organs slide across one another fairly easily. All right, so I just talked about solutions, which is basically when a molecule is going to be dissolved inside typically an aqueous media, but it doesn't have to be water. You can have another solvent if you choose, although water is the universal solvent. So we just talked about the first type of mixtures, which is the solution. This is taking a teaspoon of salt or sugar and dissolving it in water. And basically afterwards it's completely separated up into the water, so you're not able to see it, and you wouldn't be able to get it back unless you dehydrated the solution. A colloid is a specialized type of solution that's like a gel or an emulsion where it's kind of in between a solution and a suspension. It has dissolved enough that it would be very difficult to extract it back out, but it is still not entirely dissolved into solution. Something like an emulsion, right? Like when you make mayonnaise, for example, that's an emulsion. So you wouldn't be able to separate mayonnaise back out into its original ingredients, but you wouldn't really call it a solution either. And in a suspension, this is a pretty easy one for me. Picture taking sand and putting it in water. The sand goes in water, it gets wet, but it's just suspended in it. It will go up and down, but it will never dissolve into solution, right? So it's basically just going to be within the water molecules, but it's not interacting with the water molecules the way, say, sodium and chlorine will. All right, so when we talk about solutions in the lab, there's a couple of different ways that we might describe them. We might describe them by percentage or by molarity. And this is just kind of like the difference between, metric and standard in terms of like inches versus yards or feet versus yards, etc. And basically percentage is one way to look at it and molarity is another way. The percentage is going to talk about the grams of the substance suspended in a particular amount of solution in the 100 milliliters, but you can also, for example, if you have a 10% solution, you take 10 grams in 100 milliliters. But you could also put 100 grams in a liter, right? Because it's all relative as long as it's the same mass per volume. So if you go up in the amount of milliliters, you would have to go up accordingly in the number of grams of substance that you use to be able to keep the same percentage. And then the same thing with molarity. It's just another way of looking at it. You're looking at moles per liter, and you're looking at the weight of the so the molar weight of a in this case salt, right? Sodium chloride. The molecular weight of that would be 58.44 grams. So if you dissolve 1 mole, 58.44 grams of it, in 1 liter, you would end up with a 1 molar solution. Right, and if you wanted to pare that down, say you wanted to have a 0.1 molar solution, you would use 5.84 grams, for example. And don't worry, in the laboratory, if we actually get to have lab, we will end up demonstrating that for you. So we will assist you through that. Okay, so we're just about to talk about the pH scale, and before we do that, I want to kind of talk about the fact that remember when we said that water could break up into H and OH, right? Well, it's not the only substance that can release a hydrogen or that can release an OH, a hydroxyl group. So for example, hydrochloric acid can be broken down into chlorine and hydrogen. Now it's still going to be, we have one positive and one negative, so it's going to be neutral in terms of like a magnetic charge, but in terms of a pH charge, we're going to end up with way more hydrogen ions in this solution than we do OH groups. Okay? And in this case, we have a base. This is going to be sodium hydroxide. I apologize. This is potassium hydroxide. I apologize. K is potassium. So potassium hydroxide is going to be able to split up. So we have the hydroxyl group and the potassium. Again, we have no charge because we have one positive, one negative. But if you're looking at the concentration of hydrogen versus oxygen versus hydrogen versus hydroxyl group, you're going to have way more hydroxyl groups in suspension here, and that's going to be what we call basic. And we'll talk about that when we get to the pH scale in just a second, but what this is demonstrating is the difference between acids, which are going to release free hydrogens in solution and therefore increase the amount of hydrogen to hydroxyl groups ratio. That's considered an acid. And bases are going to be anything that releases the hydroxyl group so that we have more hydroxyl groups than hydrogen groups inside the suspension. And a salt is not going to release either a hydrogen or a hydroxyl group, something like potassium chloride or sodium chloride. It's still going to dissolve into it, but it's not going to change the pH. So the salt won't change the pH, but anything that has OH in it or H in it is going to change the pH. Anything with the H is generally, if we have more H's than OH, we're going to make it acidic. If we have more OH's than H, it's going to make it basic. And this is what the pH scale looks like. So I'm going to take a step back for a second and talk about some things that we would normally find in our body. So for example, the blood and our tears and our sweat, all of our bodily fluids with the exception of urine and stomach acid and vaginal juices, those are going to be here in the 6 to 7 range. It's going to be neutral. And you can picture pure water is going to be exactly even for hydrogens and oxygens. So what is this depicting? This is depicting the concentration of the hydroxyl group, and this is depicting the concentration of the hydrogen ions in solution. And it's a logarithmic scale whereby when we end up with more of the hydrogen atoms free in solution than we do hydroxyls free in solution, we end up more and more increasingly acidic. Towards the middle, we're going to be even. Towards the basic or the alkaline section, we're going to have way more of the OH than we do of the H group. Right? This is where we're going to find things like bleach and ammonia. On this side, we're going to find things like stomach acid, etc. So pH balance is one of the things that has to maintain in our homeostatic equilibrium. It's going to be very important for our body to maintain blood pH, for example. But in our stomach, we're going to have a much lower pH. So again, we're going to have to maintain pH for the region specific within our body. And how do we do that? We use buffers. So there's specialized type of compounds that are going to help regulate your pH. And that's going to help you maintain the homeostasis of your body fluids. Again, I mentioned this is really very critical for maintaining normal physiological conditions. Now, these buffers are going to be able to either convert strong acids or convert weak or strong bases into weaker acids or weaker bases. So it's going to, for example, if we have hydrogen ions free in solution, it can bind to this buffer here to become H2CO3. That's going to help remove that hydrogen ion from the bloodstream. So if the blood's a little bit acidic because it has too much of the free hydrogen ions, this will help bring it more towards either a weak acid or towards neutral to be able to pull those hydrogen ions out of solution. And they can also do the opposite if we end up with too many of the hydroxyl groups. All right, but what's special about carbon? Carbon is really important because carbon helps create all of our organic compounds and our organic biomolecules. And carbon is going to be able to make 4 different bonds, which gives it a lot of versatility in its combination of shapes that it can make. So if you picture that it can make 4 single bonds, it can make 2 double bonds, it can make 1 double bond and 2 single bonds, it could make a triple bond and a single bond. It has a lot more versatility than other atoms. Additionally, because of the way that it binds with things, it doesn't dissolve easily in water, and it's going to make a really good source of energy. So it's going to be one of those molecules that's going to store energy or release energy. It's going to create some molecules that are going to help store or release energy. Now, I don't expect you to memorize these functional groups here, okay? We are not in a basic chemistry class. But I want you to understand that we have different functional groups and that carbon reacts differently depending on what it's associated with, just like that teenager who might be an angel with one group of friends and a hellion with another. So some of the major functional groups of different organic molecules, note there's no carbon here, but these are going to be groups that might hang off of a carbon backbone. This here is an hydroxyl group. Oh, also let me take a second here. This R group is going to be the same for all amino acids, right? So this is actually looking at a region that's going to be the same. So we're just going to look at R as XYZ. It's always the same. And hanging off of this carbon molecule, we have an OH group. That's hydroxyl. Remember we talked about that when we talked about pHs. And that's going to be anything that's an alcohol is going to have that OH group hanging off of it. Anytime you see an S, it's going to be a sulfhydryl group, generally an SH. This is going to be important when we talk about proteins a little bit later because this is kind of like a Velcro region of a protein that's going to allow it to fold upon itself and interact with another region of the protein. So it's going to help stabilize the shape of the protein. This region here is a carbonyl region. Again, R is the general. It's going to be part of the amino acids. You can just picture this as like the backbone and the backbone. And this carbonyl group is going to be C double bonded O. That's going to be something like a ketone. And here we have C double bond O with an H, and that's going to be an aldehyde. And ketones and aldehydes can kind of convert back and forth pretty easily because you can see the structural similarity. It's just going to be releasing a hydrogen and picking up another, whatever that backbone is, or releasing that backbone and picking up another hydrogen. So R just means like a variable group of the carbon-based backbone. Now, a carboxylic acid can go back and forth between COOH and COO negative. And you can see if it releases that H, it means that positive is going to go with it. We're going to end up with a negative charge. And that's going to make them reactive. So we're going to see these when we talk about amino acids, because amino acids have a amino end and a carboxylic acid end, and the carboxylic acid end is going to be reactive. So all amino acids are going to have that -COOH group at one end, and the other end is going to have the amino group. And this is actually going to be able to react. If we remove this H, then we can bond it to another R group. And this is how we're going to end up with a chain of amino acids bound to one another. Another one that we have here is an ester. It's going to be COO bonded to another variable group. So it's going to be generally found in triglycerides and fats and oils. One of the common esters that we may use, maybe not anymore, but we used to use commonly as a painkiller is aspirin or salicylic acid. You might also recognize salicylic acid from your face washes and your acne relief because it does help kill bacteria, which can be a cause of acne. But aspirin is actually something that's found in the bark of the willow tree that we used as a pain relieving molecule for many, many years before we came up with ibuprofens. And another one you'll see here is this phosphate group. You're going to see POOO, right? So PO4, and it's usually 2 negative. And it's going to be found in ATP. We will talk extensively about ATP throughout the semester, and ATP is an energy carrier molecule that has a high and a low energy form. The triphosphate form is the high energy form, and DP, so ADP would be adenosine diphosphate, is the low energy form. Again, tri being high energy, di being low energy, and we have several of these that are just going to pop onto one another to make kind of a chain hanging off of the adenosine molecule, which is where we're going to be able to, again, carry our high energy bonds that might pick up energy from one chemical reaction and use it somewhere else to transfer that energy to another chemical reaction, kind of like an energy carrier molecule, like a currency, essentially. So we already talked about amino acids having an acidic group, carboxylic acid group, but on the other side of the amino acid, we're going to have the NH2 group, which is the amino group. And that's going to actually be able to become an NH3 positive. It's going to be able to pick up one more hydrogen ion, giving a little bit of a charge. You may recognize, if I go back just a little bit, that COO could become a little bit negative. So now you can see how it might react if this is negative and this one is positive. The amino group would hold hands with the carboxylic acid group, causing a long chain of what we then would call amino acids would call coming together would become a peptide or eventually a protein. Okay, so let's talk about the different types of biomolecules. The first that we're going to talk about are carbohydrates. And before I jump into this, I want to kind of talk about how we put biomolecules together. So we have something called a monomer versus a dimer versus a polymer. Mono being one, di meaning two, poly meaning many. And each of the different groups that we're going to talk about, so here we're talking about carbohydrates, but we're also going to talk about lipids, for example, have what we call a monomer, or a single compound, and then a dimer, which would be two, and then a polymer, which would be many put together. You can picture these like train cars. So a monosaccharide would be one saccharide. Simple sugar that has 3 to 7 carbon atoms is going to be smaller. Examples of those would be glucose, fructose, galactose, deoxyribose, and ribose. And you'll hear all of these extensively as we go throughout the lectures because deoxyribose is found in DNA, ribose in RNA. Galactose is in milk sugar, fructose, is in fruits. And when we put some of these together, these monosaccharides can go hand in hand, make a disaccharide. Some examples of those would be sucrose, which is a glucose and a fructose. Lactose is glucose and galactose. Maltose is glucose and glucose. So the monomeric form, or the monomer, would be a monosaccharide if we're talking about sugars or carbohydrates. The dimer would be a disaccharide, and the polymer would be polysaccharide. And poly just means many. It can be anywhere from 10 to hundreds of them all stored together. And a good example of this is glycogen or starch, and we'll talk about these kind of a little bit later on as well. Glycogen is a stored form of carbohydrates that are found in animals, found generally in like animal liver, for example, and starch is going to be found in plants. And surrounding this is going to have cellulose. So cellulose is going to be a region of the, I apologize, a third type of polysaccharide is cellulose. And cellulose is actually a very unique polysaccharide because we are not able to digest it. There's only a specialized set of animals that are able to digest it, like termites, for example, are able to digest wood. A termite walks into a bar and asks, "Where's the bartender?" I know that's not funny, but you can picture that termites are an animal that's able to eat cellulose. He would be able to eat the wood, so he's asking where his food would be tender. Anyway, cellulose is unable to be digested by humans, but does aid with the movement of food through the intestines. Think like the popcorn shell, right? It helps actually scrape things through. Anyway, as we continue through, you're going to see all of these different types of biomolecules have a monomer, a dimer, and a polymer. This is what a monosaccharide looks like. Now, I am not going to ask you to memorize anything more than the fact that sugars are going to be either pentoses or hexoses, which means they either have 5 carbons or 6 carbon. And you're basically going to be able to see glucose is the one that we're going to recognize most often, C6H12O6, and it's going to form long chains of glucose or fructose molecules. So monosaccharides are just going to be a single sugar by itself, like one train card, a monomer. An example of that, deoxyribose and ribose. These guys are pentoses. Again, they have 5. These are specialized forms of monosaccharides. They're going to be using it in DNA and RNA. So these are not generally used. This is used for the creation of protein. So DNA is going to tell RNA what to do, and RNA tells protein what to do, essentially. We'll talk about that separately as well. But my point is that pentoses are found in the nucleus generally, or in RNA, and hexoses are going to be used as your energy. So glucose can be broken down, for example, and release energy in the body. Disaccharides. This is a dimer of 2 sugar molecules. In this case, it's lactose, comprised of galactose and glucose. So where there was previously an OH group on one and an H group on other, by the process of dehydration synthesis, these two were merged together and a water molecule was released. Another example of this is maltose, glucose and glucose. If you've ever had Whoppers or malt balls, this is where that's the sugar that you'll be able to taste in that. And a polysaccharide just means long chain, right? So if you picture each one of these hexagons as glucose, they are all chained together in a very long chain. This could be a starch, it could be it could be a starch, it could be cellulose, it could be multiple different things, but it's going to be a long chain, basically, of glucose. These are polysaccharides. So we're moving on now from carbohydrates to lipids. Lipids are another type of biomolecule. We have multiple different types of lipids in the body, including fatty acids and triglycerides, phospholipids, steroids, ecanosoids, and a couple other lipids. So let's start talking about fatty acids. Fatty acids are the building blocks of triglycerides and phospholipids, and they are also going to be able to be catabolized, means broken down. So anabolic builds up, catabolic breaks down. So they're able to be catabolized to be able to generate ATP. And as I mentioned, ATP is a storage molecule for energy. So ADP, or diphosphate, is going to be lingering around somewhere where there's free phosphate and energy is released from a reaction. That energy is going to be gobbled up, and the triphosphate form is going to be able to hold that energy and go somewhere else in the body where it's going to release the energy, driving the next reaction. We also have triglycerides. These are generally going to be fats and oils used in energy storage and insulation. Phospholipids are very specialized because they're an amphipathic molecule. Amphipathic means it has a polar part and a non-polar part, if you picture like water and oil. So it has one region that interacts well with water and one that interacts well with oil. And this is going to allow it to create a barrier between the inside and the outside of the cell. So the cell membrane is going to be comprised of phospholipids, which are going to be able to keep certain things out and allow certain things through. So that's going to make a semi-permeable membrane. We'll talk extensively about steroids in its own chapter, but steroids include things like cholesterol, bile, vitamin D, adrenocortical hormones, and all the sex hormones. And these do multiple different things in your body, but basically they're going to help regulate physiological processes. Cholesterol specifically is going to be found in the cell membranes and is also going to be a precursor to the bile salts that we'll talk about in a second, which are going to be responsible for digestion of lipids. And then vitamin D is going to help with calcium regulation in the body and is going to help with bone growth and repair. Adrenocortical hormones are going to be in the adrenal cortical gland, and they're going to help regulate metabolism, stress reduction, and water and salt balance. And then sex hormones are going to help stimulate all of our reproductive functions and sexual differentiation and sexual characteristics. Some other types of lipids include ecanosoids, which are going to help with things like blood clotting, inflammation, immunity, and stomach acid secretion. And we are going to go through each of these extensively. They're going to have their own little day later on in our semester. All right, so fatty acids are going to be either saturated or unsaturated. And basically the difference between saturated and unsaturated is whether or not there are any double bonds. Is there room for any more hydrogen to pop on in here? So this one is saturated because there are no double bonds. This one is monounsaturated because there is one double bond. If there were two double bonds, it would be a diunsaturated, indicating that there was more than one region for hydrogen to sneak in there. Right? So if we were to bring in an H2 molecule, we could break that up and put one hydrogen connected to this carbon and one hydrogen connected to that carbon. That would straighten out this little tail, and it would become a saturated fatty acid. Okay? Now, if you can picture these like paper clips, for example, you could picture that the saturated would be all the paper clips that are fresh out of the box, right? And you could put a ton more paper clips inside the box if they're all flat than if you went through and put a kink in each of them and then try to put them back into the box. So the unsaturated fatty acids are going to actually be a little bit more, I want to call them slippery, because they're going to make it that there's more space in between them, because they're going to have the kinks in the tail. And that's actually going to be kind of how you can determine whether things are mainly saturated or unsaturated fatty acids. For example, baking grease is going to be solid at room temperature. It's going to be more of a saturated fatty acid. Whereas if it's unsaturated, it's going to be something like olive oil, where it's going to be liquid because it's going to be a little bit more fluid at room temperature. Now, here we have a fatty acid molecule that's going to be put in on the glycerol molecule. Here again, it's going to happen by dehydration synthesis, meaning we're going to have two different molecules coming together to become one larger molecule and lose water in the process. And that's going to be putting glycerol together on a fatty acid. Now, if we do 3 of those, right, 3 different fatty acid tails on the glycerol head, that's going to make a triglyceride. And triglycerides are very important in terms of insulation and energy. So this is going to be in one of the reserve energy storages. It's going to be, if you can picture something like whale blubber, is going to be very helpful for insulation for marine mammals and also for us humans, right? And these triglycerides are going to be held together here via an ester linkage. And again, you can see this one is saturated, this one is saturated, this one is monounsaturated. So we have one region that is not saturated. And this is actually going to be a triglyceride, or what you might know as a fat molecule. Now, if we have 2 of these fatty acids that hang off, and instead of having a glycerol head, we're going to have a phosphate head. Now we make something called a phospholipid, and phospholipids are vital. They are a major component of cell membranes, and they orient themselves in a specific direction. So as I mentioned, they have one region that's polar, right, or water-loving, and one that's non-polar, or water-fearing, right? So we have a hydrophobic region and a hydrophilic region. One area interacts well with water, one interacts well with oil or fats. And if we orient them all with the polar heads on the outside and the tails dangling, and then we make another layer the opposite direction with the polar heads this way and the tails dangling, so it's actually a bilayer, now we have a cell membrane. And in this way, we can keep certain things on the outside of the cell and let other things keep other things on the inside of the cell, and certain things are going to be allowed to pass through because we'll have channels or different regions like cholesterol and different spots where things are going to be able to fit through. Okay, so there are other things that are going to be in this cell membrane, including, like I said, cholesterol, glycoproteins, or identification molecules that are going to be found lodged in these cell membranes as well. And that's going to become important when we talk about hormonal regulation, when we talk about the endocrine system, because we're going to have molecules that are tumbling through solution that are going to have to interact with the outside of these cells to be able to become target tissues or organs, right? All right, again, I'm not going to ever ask you to memorize the structure of any of these. We're not going to be going over the chemical structure. I'm never going to say what specifically is this compound, but I do want you to understand that form defines function, and we have different forms here for different steroids that are going to have different functions. And the basic steroid backbone are these 4 rings here. Okay, 3 of them are hexagons and 1 is a pentagon. And we have a long hydrocarbon tail here that's going to make cholesterol different from its partners, from its other steroids family members. Here's estradiol, which is a female sex hormone, and it's going to be very similar, as you know, to testosterone, right? What's our major difference? We have this and that. So estrogen or estradiol and testosterone are going to be very similar. And I also want to take a moment to let everyone know here that it is not entirely sex dependent. So it's not that females only make female sex hormones and males only make male sex hormones. It's a ratio. Females make more sex hormones that are female sex hormones than males do, but they still have testosterone at a base level, and males still have estrogen or other sorts of female sex hormones at base levels, right? But it's a ratio of the two. We also have cortisol, which is a stress hormone. Cortisol is going to be involved, so you might take hydrocortisol, for example, if you end up, hydrocortisone would help with cortisol, if you end up with an inflammatory response. So cortisol is going to be a stress molecule, which is going to help with the inflammatory response. All right, so another class of biomolecules are proteins. Proteins are structural and also regulatory and also protectory, and they basically do everything within the body. Proteins have specific functions based on their amino acid sequence, and they all do exactly what they are told to do. So some of them provide protection, some of them are going to be muscular contraction, some of them help transport things, others are going to help serve as enzymes, which are kind of like biological matchmakers to help speed up the rate of reactions. So proteins do a plethora of stuff. They are the workhorse or courses within the cell. And proteins are comprised of amino acids. So as I mentioned, everything has a monomer and a polymeric form. The monomer is going to be an amino acid, and then two amino acids together become a peptide, right, because of one peptide bond, and then eventually the long chain of peptides become a polypeptide, and the polypeptide eventually becomes a protein. And this is what I was talking about. One side is going to be the amino group, one side is the carboxylic acid group, and as you may recall, it is easy for this OH group and this H group to join together to form water, right? So we will have, well, actually it wouldn't happen amongst them, it would happen two next to each other, so it would happen from one amino acid and then the amino acid next to each other would react. But anyway, we lose one OH and one H group, it would form water, and we would have a dehydration synthesis reaction occurring whereby they would bind to one another. I also want to point out that the reason that would happen is because we have we have a positive and a negative charge, and that happens when the amino acid becomes what we call doubly ionized. So we go from a non-ionized version where we have just normal COOH and NH2. Neither of these are reactive because they're not positive and they're not negative. But when N picks up another hydrogen, now it's NH3 positive, and this is COO negative. And when that occurs, these guys are going to be positive and negatively charged. They're going to end up binding to one another. We also, and that's going to happen by way of dehydration synthesis, we also are going to have different variable groups that hang off. So here's our variable group. Here's our side chain. Here's just hydrogen. It's going to be one of the smallest, simplest amino acids that's out there. It's called glycine. The reactive group here is just H. In this case, cysteine is going to have a reactive group, SH and CH2. Again, that S is a sulfur, and it's going to, as I mentioned previously, help be like a little Velcro, so it's going to allow the protein to fold in among itself. Another one here is tyrosine. It's going to have a large ring in the middle, so it's going to be much larger, for example, than glycine. And then here's lysine, which is a much longer chain. What I want to point out here is that these variable groups vary both in size, shape, and charge. And so when we talk about proteins and how proteins interact with both how they fold among themselves and how they interact with other proteins, that's all due to where these variable groups are located within the protein and how those variable groups react with what's around them. As I mentioned previously, a polypeptide chain is formed when the carboxylic acid group of amino acid A interacts with the amino group of amino acid B. And again, we're going to bond these together by dehydration synthesis. So one hydrogen is going to come off this OH group. It's going to come off, and the next thing you know, we have a peptide bond between this carbon and that nitrogen. And that happens again through dehydration synthesis because we have the release of water. The opposite of that would be the catabolic reaction, where we take this dipeptide, right? A polypeptide, we could have dipeptide, tripeptide, etc. And that is going to end up, if we break it down from a dipeptide into two amino acids again, that again is hydrolysis. So again, anytime that we are catabolizing something, it happens through hydrolysis. If we're building things up, it happens by dehydration synthesis. And just to hammer home the idea that we have a monomeric and a dimeric form of each of these biomolecules, the monomeric form here is glycine, alanine, any of those single amino acids by themselves, and when we put them together forming a peptide bond, now we have a dimer. In this case, it's a dipeptide, right? We put several of these together, we end up with a polymer, in this case a polypeptide, and eventually we have a fully functioning protein. Alright, so proteins have multiple levels of biological complexity. So what I just talked to you about was amino acids sitting next to one another holding hands, right? A next to B, next to C, next to D. And that looks like this. That's the primary structure. This is the first amino acid, bond to a peptide bond to the next amino acid, bond by peptide bond to the next amino acid in a long chain. And if you can picture this as just like a strand of hair, it's going to be a very long chain, but it doesn't have any function yet because that's just the actual order of the amino acids or the sequence of amino acids. We have to have some sort of secondary complexity. So if you can picture that the hair could end up in a curl, that would look like an alpha helix, okay, or a staircase, a revolving staircase. This is going to be an alpha helix, And what's happening is that the amino acids are not only able to interact with the amino acid directly to the left and the right of it, but also amino acids above and below it. Right? So now we're more on the level of like an apartment complex where you're able to interact with the neighbors above you as well as the neighbors to the left and the right. And so this gives it what we call secondary structure. And there's a couple secondary structures we can make. That's the alpha helix is the one thing. Another is kind of like it's called a beta pleated sheet. It's like corrugated cardboard basically. It goes back and forth and back and forth. And again, it's going to be a structured way in which amino acids are able to interact with amino acids that might not be directly next to each other. Now, these are just going to be secondary regions within the protein. The entire folding of the polypeptide chain, the final three-dimensional shape, is going to be its tertiary structure. And then if we put a couple of these polypeptides together, we can end up with a quaternary structure. One of the most classic examples, of course, is going to be hemoglobin, which has 4 separate molecules, globin molecules inside of it that come together in quaternary structure to make hemoglobin. So as I mentioned, we have the primary structure, which is just a sequence of amino acids. Then we have secondary structures, which are folding that allow amino acids to interact with other amino acids around it. But then that's not going to be everything, right? There's going to be the entire three-dimensional structure, which includes all of the alpha helices and all of beta pleated sheets. That's tertiary. And then quaternary is multiple proteins or polypeptides put together. All right. Let's switch gears for a second and start talking about enzymes. And enzymes are biological catalysts. They're basically matchmakers, and they speed up reactions that would already occur but would occur at a much lower rate. They are very specific, they are very efficient, they do not change throughout the reaction. So they might change during it, but at the end they return to their original state. And they are also subject to multiple different sets of cellular regulation. So this is what an enzyme looks like, and we'll talk about many enzymes throughout the course of this class. Enzymes generally are going to end with ACE. So if you hear something that ends with ACE, sucrase, amylase, etc., they're going to break down in this case, OS would be sugars. So if you have a sucrase, it's going to break down sucrose. Amylase is going to break down amylose. Right? Lactase is going to break down lactose. So anytime you have ACE, it's going to indicate that it's an enzyme. And the enzyme is actually depicted here as this yellow blob with the blue. That's the active site. The active site is going to bring together the substrates. In this case, we're actually having a catabolic reaction. So we have sucrose, which is the dimer, and it's going to be bringing in water as well, because remember, when we break things down, we have to break water as well, so it's by hydrolysis. So water is going to come in, sucrose is going to come in, and then the OH is going to end up on one side, and the H is going to end up on the other, which is going to cap off these little ends. The enzyme is going to force something to happen that would previously have happened, but at a much lower rate, right? They could possibly have broken apart, but it wouldn't have happened as immediately as the enzyme forcing it to happen. So it's going to catalyze the reaction and transform what we call the substrate, so previously that was the sucrose and water, into the products, which are now glucose and fructose. Now, although this active site and the enzyme might change a little bit during the reaction itself, they will revert back to their unchanged state, and they will be able to catalyze the next reaction using, so basically the same reaction, but using new substrates. And it's going to happen over and over and over. So an enzyme is going to be able to catalyze the same reaction repeatedly. But I want to make it clear here, it can only catalyze this reaction. Like a lock and a key, the specific shape of the active site will only let specific substrates in. And so it's only going to be able to catalyze a very specific reaction. So enzymes are very specific about the reaction that they catalyze. All right, so let's talk about nucleotides for a second. Nucleotides are like DNA or RNA, and they're basically going to be the powerhouse of the cell. So inside the nucleus, which is an organelle, the largest organelle within the cell, you're going to have DNA. And DNA is going to be basically the boss's office. It forms a genetic code inside the nuclei, and it regulates all of the cell's activity by creating RNA. So DNA has the code for RNA, and RNA has the code for the proteins that are going to be formed. DNA has 4 major bases: adenine, guanine, thymine, and cytosine. What you need to know is that they pair by size and by charge. So we need one large and one small, and they have to have the same, and when we say charge here, it's going to show us a little semicircle versus a square plug or rectangular plug. So A can only bind to T because we need one large and one small, and they have to have the same fit. And then G can only bind to C because, again, we need one large and one small, and they are going to have a different amount of of hydrogen bonding, basically. So you can see G and C are going to have 3 hydrogen bonds, and A and T are going to have 2 hydrogen bonds. And that's how we have specificity in this molecule. The other thing that's interesting is, as you recall, hydrogen bonding is not, it's very strong overall, but every individual one is weak. So we are able to open this up or unwind this DNA and put this DNA back together, kind of like a Velcro or a zipper, fairly easily because of the fact that the hydrogen bonding is not the very strongest bond in the entire set of bonds, right? So just to compare DNA and RNA, DNA and RNA are both made of nitrogenous bases, but there is one that differs between DNA and RNA. So in DNA we have A, T, C, and G. In RNA we have A, U, C, and G. So we have uracil instead of thymine. We also have a different sugar. So deoxyribose has 1 less oxygen, deoxy, than the original ribose. And additionally, DNA is double-stranded and RNA is typically single-stranded. So DNA, as I just showed you, was a twisted helix. This is actually a separate strand here than this one. In DNA, A binds with T. In RNA, A binds with U. In both cases, G is going to bind with C. And what's depicted here is the number of hydrogen bonds. So as I mentioned, it's by size and by charge. So 1 large, 1 small, and 1 with 2 hydrogen bonding, and 1 with 3 hydrogen bonding. DNA is self-replicating. That means that the new copy of DNA comes from an old copy of DNA, whereas RNA is actually made from DNA as a blueprint. So DNA does both things. It replicates itself, and it also creates RNA. And what does it do? It encodes information, again, both for self-replication and also for making proteins by the form of creating RNA, and then RNA is going to go out into the world, carry that genetic code out, so outside of the nucleus, into the cytoplasm, and make proteins. We have 2 different types of DNA, nuclear DNA and mitochondrial DNA. You may know a little bit about the 7 daughters of Eve, but anyway, all of the mitochondrial DNA in your lineage comes from your mother because the mitochondria in the sperm are actually lost during fertilization. So the mitochondria never make it into the egg, and that means that you're only able to detect your maternal lineage through mitochondrial DNA. RNA, we have multiple different types of RNA, and these are all going to be used in the creation of proteins. So we have messenger RNA, transfer RNA, ribosomal RNA, and we'll talk about that extensively when we get to transcription and translation. All right, so adenosine, you may have just seen 1 second ago in the DNA, right? You have adenine here, and adenosine is created with adenine and then the ribose, and then we can have either 2 or 3 phosphate groups. You can also have 1, it would be monophosphate, but we don't really talk about that much. So here's adenine, ribose, and the phosphate group. So basically adenosine is made of a sugar, the adenine, which is the nitrogenous base, and then either 2, which would be the lower energy form, or the 3rd phosphate would give it the high energy form. And 2 would be diphosphate, adenosine diphosphate, and 3 would be adenosine triphosphate. And as you go from ATP to ADP, you lose energy, and as you go from ADP to ATP, you have to use energy. You're going to be gaining energy, basically taking energy from your surroundings. So basically adding this on takes energy and is going to store it, breaking it off releases energy, and is going to be able to use that energy for another reaction. Okay, so I appreciate you slogging through that with me. I know that is a whole handful, and we touch on multiple different topics that would normally be their own lecture, but because we need to focus on the anatomy side of things, I just kind of wanted to gloss over on the chemistry. But I really do appreciate you sticking with me all the way to the end there. Thank you very much, and happy studying. Aloha!